So examples are lotus and rose for apocarpus, mustard and tomato for syncarpus. Okay. Next, so based upon how these ovules are placed on the placenta and what is the shape of this placenta, we are again dividing dividing the ovules into these many types. So arrangement of ovules on the placenta within the ovary. Okay, ma? Based on that, placentation is of how many types? 5 types. One is marginal, second one is axial, parietal, free central, and the fifth one is called basal. Able to understand now? So this is called as marginal, this is called as axial, this is called as parietal, this is called as free central, and this is called as basal. Able to understand now? Now let us study the arrangement of these ovules on the placenta. So first of all, we are discussing about marginal arrangement. Marginal arrangement means what? In the figure only you can see, no? Okay, so this is the placenta, and to the placenta the ovules are arranged just like in a margin, in a straight line. Okay, ma? So this type of arrangement you can call it as marginal type of arrangement. So here a placenta forms a ridge, okay, ma? Along the ventral suture of the ovary, and ovules are born on this ridge forming 2 rows. Are you able to understand, ma? So placenta, it gets divided into 2 parts, ma? On which side? On the ventral side. Okay, and on that ventral side, these ovules, they are getting attached to the placenta. Are you able to understand? So what is the example of this one? Example of this one is P. Okay, so 2 rows of arrangement you can see. Okay, so on one ventral suture you can find one row of ovules. On the another one you can find the second row of ovules. Clear? So when you open this P seed, then P, shall I call this one as fruit? Vegetable is fine, but shall I call it a fruit? Fruit only, no, basically this one. So if you open this one, then this is the type of seeds which you see, Noma, and that is the exact arrangement of ovules inside the placenta. Fine, that's it. Clear? So next, axial. What is meant by axial? So in the figure only you can see, no? So these lines which you are seeing here, they are called as septum. Septum, singular form. Septa is the plural form. It means the fruit is divided into some parts. So for example, if you cut a line, lemon, lemon, sorry. If you cut a lemon, you can see septum present in that, no? Divisions. Are you able to understand? They are called as septum. And to the placenta, if the ovules are arranged in this type of arrangement to the central region, then it is called as axial type of arrangement. Are you able to understand? So placenta is axial. Okay, it means it is passing like an axis. Okay, and ovules are attached to it in a multilocular ovary. What is meant by multilocular ovary? So this total you can call it as one locule. Okay, this one you can call it as one locule. This one you can call it as one locule, or you can call it as septa also. Septum is the plural form, singular form. Septum is the, septa is the plural form. Clear, Nama? So examples of this one is what? China rose, tomato, and lemon. Okay, ma? So are you able to see these divisions? Divisions are present here, Nama? So all the ovules, they are not just arranged like this. There are some divisions also, and among the divisions you can find these ovules. Okay, and what is the arrangement of the placenta? It is axial. It is like behaving like an axis to the fruit, and to the length of the fruit these ovules are arranged. Best example is tomato, Nama. If you cut a tomato, then this is the type of arrangement which you can see, no? So this is the septa which are separated. Okay, and this is the placenta which you are seeing here. Okay, and to the placenta you can see the seeds or ovules which are attached like this. Fine, that's it. So examples of axial type of arrangement: China rose, again you got China rose here, tomato, and lemon. So these are the examples. Clear? So this is an example of a lady's finger. Are you able to understand? No? Here also you can see this type of arrangement, and see how ante మీ వేళ్ళు కోస్తే ఉంటాయి కదా కోసుకుంటారు ఇంటికి పోయి మళ్ళీ. Ladies ladies finger is a vegetable. Clear, Nama? So these are many septa which are present here. Are you able to understand? And in each septa, in one row, you can find only one ovule to be present or one seed to be present. Fine, ma? That's it. Okay. Axial. Already we have studied this one now. So next one is parietal. By looking at the figure only, I can see now what is meant by parietal type of arrangement. Clear? So here the ovules develop on parietal type of placentation. So ovules are developed on the inner wall of the ovary itself, or you can call it as the peripheral part. Are you able to understand, ma? So here, this is the development of the ovules inside the ovary. Clear, ma? So ovary is one chambered, ma, but it appears like it is a two chambered because of a development of a false septum. Septum means separation. False separation means it appears as if there is a separation, but actually there is no separation. So that is called as a false separation. So examples of parietal type of placentation are mustard and archimon. Clear? Next. So free central. So here ovules are born on the central axis, and septa are absent. Okay. So what is the difference between the previous condition and this condition, ma? In a parietal type of placentation, all the ovules they were attached to the peripheral part, or you can call it as the circumference part. Clear, nama? But here all of them they are attached to the central part. Clear? Are born on the central axis, and here there is no concept of septa. Separations are absent. Clear, no? So in free central type of placentation, separations are absent. So this is one important point. And examples are dianthus and primrose. So these are the two examples of free central type of placentation. Okay, ma? Next, the last one, which is called as a basal placentation. So what is meant by basal placentation? Here, placenta develops at the base of the ovary itself. Clear, Nama? And you can find only single ovule will be present. Okay. So where you can find this placenta, you cannot find this placenta as an axis. So generally, in all the remaining type of placentation, placenta was present like an axis, Nama, and to the axis, all these ovules they were attached. But here, what is the concept? The placenta develops at the base of the ovary itself, and there is only single ovule which is attached. Are you able to understand, ma? So this is the structure of placenta, which is developed at the base of the what you called ovary. And examples of this one is what? Sunflower and marigold. So what is the unique point of basal type of placentation, ma? This is the unique point. There is only single ovule, only one ovule will be present. It means in this total fruit, you can find only one seed will be present. That is the unique nature of basal type of placentation. Clear, ma? Next, after fertilization, the ovules develops into seeds and the ovary matures into a fruit. This already we have studied, no, ma? So inside the gynecium, the ovary itself that develops into what? That develops into the fruit. Okay, and the ovules which are attached to the placenta, they will become the seeds after fertilization. Are you able to understand, ma? So if fertilization doesn't happen, then also fruits can develop, but they will become seedless fruits. Are you able to understand, ma? If fertilization happens, ovules will develop into seeds. If there is no fertilization, then also fruit will develop, but seeds will be absent. There is no concept of seeds. Are you able to understand, ma? So these ovules, they will disappear. Okay. So that type of that type of development of the fruit, I think it is called as a parthenogenesis. Clear, no, ma? So we will study later. Okay. Now see here. See this, ma? How this flower it has developed into a fruit. Okay. Now here you can see the stigma, no? Stigma to the style. You can see the stigma, no, ma? Clear? That stigma where it will be present, it will be present at the end of this fruit. So here you can find a dot-like structure on a tomato, no? What is that dot-like structure? That dot-like structure is nothing but the enlarged, what you call the stigma. Are you able to understand, ma? So therefore, here you can find this ovary, no, ma? So this ovary itself it develops into what? Ovary itself it develops into a fruit. Clear, no, ma? And inside this one, you are able to see this ovules, no? Ovules. So these ovules only finally they develop into seeds. So this is what is going to happen. Clear number? And pedicel is common to both of them because the same flower only develops into a fruit. No? So first of all, what is the supporting structure of a flower? Pedicel. To the pedicel, what is attached? Thalamus is attached. Then the different what you call appendages of the flower will start. Clear number? So same pedicel is common to flower also. Same pedicel is common to fruit also. Similarly, you can find the green colored parts which are called as sepals. Number. So this fruit also will have the same sepals. Okay. And what happens to these petals in the process of fruit formation? These petals they get dried up and they will fall away from the flower. Are you able to understand more? And the main ovary it develops into the fruit. Ovules will develop into seeds. That's it. So that is what is happening in the case of fruit formation. Clear now? Okay. So next we are studying about the fruit and the seed. Clear? Okay, ma? Now, sir. So, what is meant by fruit, ma? It is a ripened ovary developed after fertilization. After fertilization, whatever structure it develops into a big structure, that is nothing but fruit. So, therefore, fruit can also be called as a ripened ovary. Ovary, when it ripens, that is also called as a fruit. Clear? So it is the characteristic feature of what? It is the characteristic feature of flowering plants. Clear? So a fruit formed without a fertilization of ovary is called as parthenocarpic fruit. Okay, ma? So what is meant by parthenocarpic fruit, ma? Fruit will develop without fertilization. It means ovules, they are not developed into seeds. So that type of development is called as parthenocarpic fruit. And these fruits are seedless, no? They doesn't have any seed. Okay. And mango and coconut, fruit is called a drupe. So in the case of mango as well as coconut, so generally these are also fruits only, but what is the general name given to them? They are called as drupes. Clear? So they are one seeded and develop from monocarpellary superior ovaries. So what is meant by monocarpellary? So inside the flower, inside the gynoecium, you can find many carpels. No? Many carpels may be present. Are you able to understand? But this mango and coconut, they are formed from how many carpel flowers? Single carpel will be present. Are you able to understand? Ma? It means there is a single ovary, there is a single style, and there is a single, what you call that one? Stigma will be present. Are you able to understand? Ma? So monocarpellary flowers only, they get from this mango and coconut, they are developed. Clear? And they are containing of how many seeds, ma? They contain only one seed. So that is a speciality of mango as well as coconut. Clear? So basically we can divide the fruit into two parts, ma. Total fruit you can divide into two parts. What are the two parts? One is called the pericarp and the second one is called the seed. Okay. So total fruit you can divide it into only two parts. One is the pericarp and the second next one is called the seed. And pericarp is again divided into three parts. Are you able to understand? So pericarp, it may be dry or it may be fleshy depending upon the nature of the fruit. Clear number? Pericarp is also called as a fruit wall. Clear? So thick and fleshy pericarp is differentiated into outer epicarp, inner middle mesocarp, and the inner endocarp. Clear ma? Epicarp is also called as exocarp also. Are you able to understand? Okay ma? So the wall of this fruit, which is called as a fruit wall, or you can call it as pericarp, it can be of two types. It can be dry also, it can be fleshy also. If it is fleshy, again it is divided into three parts. Are you able to understand? If it is dry, it is not divided into three parts. If it is fleshy, then it is again divided into three parts. So what are the three parts? So outer epicarp, outer layer. You should have seen the outer surface of the mango, no? Skin will be there, no? That is called as exocarp, or you can call it as epicarp. Are you able to understand? Skin of the mango. And inside the skin, there will be a fleshy membrane which we eat, no? That is called as mesocarp. The fleshy membrane which we use for eating, that is called as mesocarp. That is the middle layer of the fruit wall. Are you able to understand? No, fruit wall only we are dividing into three types. Don't forget here. Fruit wall doesn't means only skin of the fruit. No, fruit wall means that endocarp, epicarp as well as mesocarp. All the three combined it is called as fruit wall. Okay. So skin of the mango also comes under epicarp. Pericarp. Are you able to understand? There is a fleshy layer which is called as mesocarp. That also comes under fruit wall. Are you able to understand? And when you totally complete the mango, there will be hard layer inside the mango, Nama. In that hard layer, again you will find the seed. Are you able to understand? That hard layer, what you call that one, that is called as the endocarp. So these are the three different parts of what fruit wall or pericarp. Okay, ma? And pericarp is differentiated into three parts in which condition? Only if it is a fleshy. If it is a dry, there is no concept of three parts, only one layer is present. Are you able to understand? That's it. Clear, Nama? So therefore, see the similarities which are present here. So here we can see very clearly. So skin of the mango, that is called as epicarp, or you can call it as exocarp. Skin of the mango. Clear, Nama? Fleshy layer, we can call it as mesocarp. And the hard layer surrounding the seed is called as endocarp. And inside this endocarp, you will find the seed of the mango. Are you able to understand? So these are the different parts. So similarly, you can differentiate them in the case of this coconut also. Okay. So skin of the coconut is called as exocarp, or you can call it as epicarp. Are you able to understand, ma? See, ma, there is a difference between the mesocarp of mango and the mesocarp of coconut. Mesocarp of mango is edible. You can eat that one. But the mesocarp of coconut, I can't eat. Maybe you can eat. Are you able to understand, ma? So mesocarp of coconut, it is completely made up of indigestible cellulose fibers. Are you able to understand? వల్చేస్తాం చూడండి పైన లేయర్ అంతా ఆ వల్సిన లేయర్ నే మనం ఏమంటాము అదే mesocarp అంటాం. But the same mesocarp is edible in the case of mango. Clear, Nama? And then you will see a hard shell will be present, Nama. A hard shell will be present in this inside this mesocarp. No? That is called as what you call that one, that is called as endocarp. Okay. And inside the endocarp, you will find this meat of the coconut, or you can call it as the meat ane antaru ante inka. Clear, ma? So these are the three structures which are present in the case of a fruit, or you can call it as a pericarp, which is a combination of three layers. Okay. And inside this endocarp, what you find inside the endocarp, you will find the seed. Okay, ma? So this is how you can differentiate a fruit. Next, he is giving something about mango and what you call coconut. Already we have discussed, Nama, so no need of studying once again. So next we discuss about the seed. Okay. So it is the mature ovule developed after fertilization. Already we have discussed, Nama. When ovule undergoes fertilization, then ovule itself it develops into a seed. Okay. Seed is made up of seed coat and an embryo. Okay, ma? Basically inside the seed what you have, it is basically made up of seed coat, and a seed coat again has two layers that we will study later. So seed coat is present and basically an embryo is present. And above the embryo or surrounding the embryo, endosperm may be present, endosperm may not also be present. Clear, Nama? So what is meant by endosperm? So that tissue layer which gives nourishment, which gives feeding to the embryo is called as endosperm. So endosperm may be present in some seeds, endosperm may be absent in some seeds. So therefore basically seed, I will say that basically it is made up of two parts. One is the seed coat and the next one is the embryo. Endosperm may be present, may not be present. Clear, Nama? So therefore it is made up of two parts. And what about radical? What about embryo? Embryo is made up of radical and embryonal axis is present, and two cotyledons or single cotyledon may be present. So seeds basically you can divide them into two types, Nama. Monocotyledonous seeds are also present, dicotyledonous seeds are also present. Are able to understand? If you can divide the seed into two equal halves, then it is called as dicotyledonous seeds. If there is only one cotyledon, then it is called as monocotyledonous seeds. Clear, Nama? And whatever it may be, radical is also always present, plumule is also present, embryonal axis is also present, whether it is monocot or whether it is a Dicot. Are you able to understand, ma? So this is an example of a dichotosy. So here you are seeing two cotyledons are present, no? Two cotyledons are present. Okay, ma? And this is an example of a monocotosy. So there is only one cotyledon which is present. Are you able to understand, ma? Two cotyledons are, if there, it is called dicotyledon. And if there is only one cotyledon, then it is called as monocotyledon. Okay, ma? And what is meant by plumule and radical already we have studied, no? Clear, no? When you sow this seed inside the what you call the soil, then it starts the germination. So once it starts the germination, plumule travels upwards. Plumule travels upwards and it becomes the stem of the plant. Radical moves downwards and it becomes the root of the plant. Okay, ma? And the axis which you are seeing here connecting this plumule and this radical, that is called as embryonal axis. Okay, ma? So embryonal axis, it will be present in dicot seeds also as well as monocot seeds also. In dicot seeds, the length of the embryonal axis is bigger. But in the case of monocot seeds, here you can find the embryonal axis. In monocot seeds, this is the placement of the embryonal axis. Are you able to understand, ma? And it is very small. Are you able to understand? So basically, embryo is made up of what? A radical is present, plumule is present, embryonal axis is present, and cotyledons may be one cotyledon or two cotyledon. Clear, no, ma? Single cotyledonous seeds are what? Wheat and maize. Okay, ma? Wheat and maize, they are monocotyledonous. There's total wheat is made up of only one cotyledon. No? Clear, no, ma? Whereas grams and peas, బఠానీ రెండు పార్ట్స్ గా డివైడ్ చేయొచ్చు కదా మనం. సో గ్రామ్స్ అండ్ పీస్ దే ఆర్ dicotyledonous type. Clear? Now each of this what you called structures or parts of this seeds, we will study each of them in detail. And endosperm, what is meant by endosperm? Here I have written, endosperm is a tissue that surrounds and nourishes the embryo in the seeds of angiosperms. Clear, nama? So it is a tissue, ma, which feeds the embryo or which nourishes the embryo, which makes the embryo to be alive. That is called as endosperm. Clear? Now see. So the outermost covering of a seed is called seed coat. For any seed, you will have outer covering, no, which is called a seed coat. And a seed coat is again divided into two parts. What are they? So outer testa and inner tegmen. Clear, ma? Outer testa and inner tegmen. So these are the two parts of which seed coat is made up of. Testa is on the outer side, tegmen is on the inner side. Testa is generally dry, tegmen is generally wet. Clear, no, ma? So on the seed coat, there is a scar called hilum through which the developing seeds are attached to the fruit. Are you able to understand, ma? So here you are seeing this small structure, no, ma? This structure is called as hilum. Okay, ma? Hilum. So what is meant by hilum? So this is the region where the seed is attached to the fruit. Seeds fruit ki attach ayi untayi kada. So akkada oka mark padutundi. That mark is called as hilum. Okay, ma? And above the hilum there will be a small hole which is called as micropyle. Hilum paina malli em untundi. Above the hilum there will be a small pore, small hole which is called as micropyle. Clear, nama? So these are some of the parts of the seed. The outer part of the seed which is called as seed coat. Okay, ma? So seed coat is made up of testa and tegmen. So it has a hilum from which it is attached to the fruit, and there is a small pore which is called as micropyle. Okay, so remaining parts we will study in the upcoming this one. Okay, ma? So within the seed coat, so we are discussing about which kind of seeds, ma? Here we are discussing about dicotyledonous seeds. Don't forget about this one. We are discussing about which type of seeds, ma? Dicotyledonous seeds. Then we will go for monocotyledonous seeds. Clear? So in dicotyledonous seeds, within the seed coat there is an embryo consisting of an embryonal axis and two cotyledons, they are present. So cotyledons are fleshy and full of reserve food materials. Okay, ma? Which is also called as endosperm. Okay, ma? The food material liquid is called as what? It is called as endosperm. So cotyledons are completely fleshy, ma, and they are stored with food materials. So at the ends of embryonal axis, radical and plumule are present. That also we have discussed, ma? Radical will be present and a plumule also will be present. Okay. Now see here, some structures we are supposed to understand here. So these are the two cotyledons. A seed you can divide into two cotyledons, and this is a radical which develops into the root, and the structure which is supporting this radical is called as hypocotyl. This is an important bit. Are you able to understand, ma? So radical is supported by what? Radical is supported by hypocotyl. It means the root is supported by hypocotyl. And testa and tegmen already we have studied, seed coat. Clear, nama? And this structure from which initially leaves will develop, that is called as a plumule. Clear, nama? Which moves upwards. And plumule is supported by what? Plumule is supported by epicotyl. Clear, ma? Hypocotyl supports the radical. Epicotyl supports the plumule. Clear, no? So these are some of the parts which you should remember. Okay. Next is, so in some seeds such as castor seeds, endosperm is formed because of double fertilization. Clear, nama? It is a food storing tissue. Already we have discussed, you know. So castor is an example of endosperm which is formed because of double fertilization. What is meant by double fertilization, you know very well, no? Clear, nama? Pollen grains, they come and settle on the stigma. Pollen tubes develop. Pollen tubes goes to the base of the ovary, and inside this pollen tube, two sperms will be present. One sperm fertilizes with one thing, another sperm fertilizes with another thing. That already we have discussed in the previous classes, nama? So that concept is called as double fertilization. So in the case of castor అంటే ఏమని ఆమ్దం పెట్టుకుంటాం చూడండి తల, that is made up of castor seeds only. Are you able to understand, ma? Have you not seen castor? So if you what you called grind this castor, immediately you can see the stickiness of this castor oil, and the smell also comes out. Smell will be very beautiful. Clear, nama? So in castor, what kind of endosperm is present? Endosperm which is formed because of double fertilization is present. Okay, ma? Next, in plants such as bean, gram and pea, the seeds are non-endospermous. What is meant by non-endospermous? Endosperm is absent. Clear, nama? It means there is no nourishment which is provided to the embryo. So endosperm is absent. In which of them endosperm is absent? Bean, gram and pea. There you don't find any endosperm. Whereas in the case of castor, endosperm is present because of double fertilization. Clear, nama? So these are the diagrams of castor seeds, beans and grams. Okay. So what is meant by double fertilization? Here I have given, ma. So if you are interested, once you can go through this total data. Okay. Next. So generally monocot seeds are endospermic. So this is a very, very important bit. Clear, nama? Generally means what? It means monocot seeds can be non-endospermic also. That is the meaning of the term. No? But generally monocot seeds are endospermic. It means if there is only one monocot, one cotyledon is present, then most of them they are endospermic. Endosperm will be present. Clear, ma? But some of them they are non-endospermic also. And what is the example given? Example is orchids. Orchids is monocot, but still it is non-endospermic. It doesn't have any endosperm. Clear, ma? In cereals like maize, the seed coat is membranous and generally fused with the fruit wall. Are you able to understand? But this is another exception which you should study. Cereals means we don't know. Pulses, cereals, clear, nama? So the seed coat, the coating which is present to the seed, it is not made up of two layers. It is just membranous. Membranous means what? A very thin membrane type of layer will be present, and it will be fused with the fruit wall. Are you able to understand, ma? So it means two layers. What are the two layers of the seed coat? Tegmen and testa. So this testa and tegmen, they are absent. Are you able to understand, ma? So total seed coat is made up of a single membrane, and it will be fused with the fruit wall. Okay. So if you carefully, if you can remove that what you call seed coat, you can remove that seed coat also. Clear, no? So where you will find this membranous type of seed coat, ma? In cereals. In cereals. That is one important point. So generally endosperm is bulky and it stores the food material. Now we are discussing about monocotyledon seeds. Don't be confused about that. We are discussing about which kind of seeds, ma? Monocot seeds. Clear? So outer covering of endosperm separates the embryo by a protein layer called alluvion. What is the pronunciation of this one? Alluvion layer. Clear, no, ma? So therefore, what is the meaning of this one, Sima, here? So this total layer, this we can call it as the endosperm. Okay. So here the total food material required for the embryo is present. Are you able to understand? And this endosperm, it is separated to the embryo by which kind of layer? So here you are seeing this alluvion layer, Noma. So this layer is a layer which is made up of protein. And this layer, it separates the endosperm and it separates the embryo. Are you able to understand? That's it. So this is one point which you should remember. So what is that layer which separates the endosperm and embryoma? Alluvion layer is the layer which separates them. Clear? So next here you can see, ma, this is scutellum. Okay. What is meant by scutellum? The outer covering to the embryo is called as scutellum. Are you able to understand? So covering to embryo is called as scutellum. Clear, number? So this total part which you are seeing here, this is the embryo. Okay. And this remaining part which you are seeing here, this is called the endosperm. Are you able to understand? And both of them, they are separated by which layer? Alluvion layer. Clear? Next, coleoptile. What is meant by coleoptile, ma? So this is a sheath covering plumule. Okay. Plumule will be there, no, ma, which grows at system. So that plumule, it will be present in a layer. It will be present in a what you call a series of cells, and that sheath is called as coleoptile. It gives a protection to the plumule. Are you able to understand? So this is the plumule. Are you able to understand, ma? Next, coleorrhiza. What is this coleorrhiza? It is a sheet which encloses the radicle. So embryonal axis is made up of two parts, no, ma? The upper plumule and the lower radicle. So embryonal axis connects these two. Are you able to understand? And this plumule is protected by coleoptile and radicle is protected by coleorrhiza. So these are the sheets which protects this plumule and which protects the radicle. Are you able to understand? That's it. Clear, Nama? And see here the different parts which are already we have discussed. So this is the embryo, and surrounding the embryo you can find the endosperm separated by what kind of layer? Separated by a layer called alluvion layer. Okay, ma? So this is the coleorrhiza which gives a protection to the radicle. So this is the plumule here, and plumule is covered by coleoptile, and you can find the scutellum. Are you able to understand? Scutellum means what? It is that sheath which completely encloses the embryo. That is called a scutellum. Are you able to understand? So these are the different parts which are present in monocot seeds. Now let us see the description of each of them. Sima, already we have discussed this. So let us see. So embryo is small and situated in a groove at one end of endosperm. It means on the total top region what is present? Endosperm is present. And below the endosperm at a small region, embryo will be present. Clear, number? That's what we have discussed now. It consists of one large and shield-shaped cotyledon called as scutellum. Are you able to understand? So embryo consists of what? A layer which is called as scutellum, a shield which is called as scutellum, and a short axis within a plumule and a radicle. So here also there is an axis, just like dicot seeds. But in dicot seeds, the length of the axis, the embryonal axis, will be bigger. But here, the length of the embryonal axis will be very small. So at both ends of the embryonal axis, what you find? You find a plumule and you find a radicle. Clear, no? Plumule is protected in a sheath called coleoptile. Already we have discussed, no? And radicle is protected by a sheath called coleorrhiza. Okay, so these are the structures of the seeds. Okay, ma? So the next concept, which is called a semi-technical description of typical flowering plant, we will discuss tomorrow. Because there are some shortcut techniques to remember this one, ma.